Hello, Assalamu alaikum. Myself, Dr. Farzana W from Department of Chemistry, J.A.T Arts, Science and Commerce College, Malegaon. Welcome all of you. Today, we shall discuss about the study of chromatography or study of chromatogram. A chromatogram is nothing but a plot of detector response, that means a signal against time. A chromatogram consists of a series of peaks. Each peak corresponds to individual component detected in the sample. The function of the detector is to sense and measure the small amount of the separated component present in the carrier gas stream leaving the column. The time corresponds to appearance of apex of each peak is the retention time. It is designated with the help of letter Tg or Tr for a particular component. Number of peak obtained indicate the number of components present in the mixture. What is meant by retention time? Retention time is nothing but the time required for the component to come out from the column after injection of the sample. That means it is the time in minute between when the sample is injected and the time when the peak is recorded. Retention time are used for qualitative identification of the peak, and for quantitative analysis peak area are to be determined. Characteristic of retention time. It is the characteristic property of each component like that means under the given experiment condition it is represent the chromatographic behavior of each component of a mixture. It represents the maximum time of emergence of the peak after sample injection. It is used for qualitative identification of the component of the given mixture.